Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Rita Austin, PhD, PhD out of University of Oklahoma, but she tells me now at the moment she's in Colorado. We're going to be talking about ancient DNA and museum collections and things like that. So, Rita, thanks for coming.
My pleasure. Thanks for having me.
Yeah, you sound like a medical or a biological archaeologist, but tell me about yourself. What do you work on?
Yeah, so, that's not terribly far off, but I primarily work in museums, looking at past human remains from all over the world to try and understand past human experiences and disease processes, particularly related to tuberculosis and syphilis.
Oh, yeah. Like, how long has tuberculosis and syphilis been around, do you think? Like thousands of years?
Tuberculosis is ancient. Like, we have been evolving with it and to it, etc., for millennia. Syphilis is much more recent, as the documentation will say. It was first documented in 1494, I believe, in Naples, when it showed up in Europe. But it has very strongly or closely related subspecies of Treponema that are much more ancient and are not venereal. So it's a little bit of a mystery at this point, but basically syphilis is much newer than tuberculosis.
What are the old names for these conditions? Because I guess one is like consumption or something, and I don't know. The old names are pretty cool.
Yeah. So consumption, the wasting disease was also used for tuberculosis. But it's sort of ironic because they called it the wasting disease, but then it sort of became like a fashion statement because you got so skinny. And for many high, like noble and aristocratic Victorian people, especially women, were very, they wanted to catch tuberculosis so that they could lose the weight. So there's a lot of beauty standards associated with tuberculosis. But to your question, syphilis has also been called the Great Pox. It's been called luz. And let's see what else.
It's horrible.
Yeah. No, it's wild.
Let's talk about tuberculosis first. What? Well, thank God I've never had it. But for people that don't know, how widespread is it today? Is it still a big problem worldwide? Is it a third world type of thing? Like, what do you know about it?
Yeah, I can speak to that fine. It's so tuberculosis is caused by a bacterium called Mycobacterium tuberculosis. It is worldwide. There's different strains of the disease that are particular to different regions of the world, but overall they're basically the same. And it's believed today that about 1/3 of the world population has latent TB, so they aren't showing any symptoms, but they are infected with the Mycobacterium tuberculosis, which can then become reactivated at any time. So that's really the danger, is that you don't know that you have it, and then it could potentially become reactivated, and then you're contagious to other people. And this isn't just in third world countries. We're still dealing with it in the United States, but to a much less extent than other parts of the world, primarily outside of Europe, in Africa, Asia, and the South Pacific. But there's plenty of tests for it today. It is a curable disease, albeit with some very intense antibacterials, and it takes a long time, but the good news is that it is treatable.
What does it do to people, and why?
Yeah, so typically what happens with tuberculosis is that it will invade the body. In pulmonary or lung tuberculosis, it will invade the body through aspirated droplets. So sneezing, coughing, just general close contact with someone who's infected, and get into the respiratory system, where it basically attaches to the lung lining and walls. But the human immune system does a pretty quick job of quarantining it. So it will encase these mycobacterium cells in nodules, very dense packets of tissue, where it basically goes into like a latent period where it's just sort of waiting for the human immune system to not be so strong. And that's when it becomes reactivated and will then spread systemically throughout the body and create very damaging results to the lungs and to other tissues of the body. Skeletally, we look for specific skeletal lesions on the spine, because that's really close to the lungs and where the bacterium likes to go. But it creates a lot of cell destruction in people. So there's a lot of like unable to breathe, which is a big issue, as well as because of that, they're not able to breathe, they're not able to eat. They lose weight, and they're very, very weak, typically when they reach active stages of the disease. And it becomes very difficult to care for yourself and to live at that point.
It's crazy.
Mm-hmm.
So, when you find evidence of it, I mean, in order to do your work, do you have to look at, let's say, recently deceased people from a given disease, and that informs you what it may look like in people that have died, hundreds or thousands of years ago with the same disease? Like, how do you learn what you know? I guess that's a pretty broad question.
Yeah, no, and so what my research is really interdisciplinary, where I take molecular viewpoints as well as morphological and skeletal and archival information to try and recreate these past disease processes. Before we knew so much about the diseases, in skeletal remains, yes, you would be primarily going to like anatomical collections where there was known causes of death, and essentially analyzing the human remains to try and identify consistent lesions or patterns that were associated with this disease, tuberculosis, for instance. And then you would be able to know for sure that if you were looking at an individual that you didn't know the cause of death, that they died from tuberculosis. Unfortunately, when we look into the past, though, there's a lot of diseases, including syphilis to an extent and tuberculosis, but primarily in short infectious diseases like pneumonia, cholera, the flu, that will not leave skeletal lesions. And so we can't diagnose these specific diseases in the past because we don't have any evidence of them. But we have gotten really good with syphilis and tuberculosis, looking at that. And then with molecular methodologies, we're able to try and confirm what we think is happening or had happened skeletally, or based on archaeological context. So does that answer your question?
Yeah, why do this, though? I mean, what good is it for you to say, this person died of tuberculosis or syphilis 10,000 years ago?
Right.
100 years ago. I mean, I could see if they were an important historical figure, like, let's say you had Al Capone's body and, legend has it he died of syphilis, and you wanted to confirm that. That makes total sense. But, what if you had like some random person that no one knew who they were? They lived 4,000 years ago, and, why would it be important to see that they died of tuberculosis or something, for instance?
Yeah, thanks for the clarification. The benefit of doing this type of research is so that we can better understand these pathogens in the past and how they've changed over time, especially because these diseases are still affecting human populations today. As recent as 2015, there was an outbreak of syphilis in Oklahoma City. There's still continual outbreaks of TB around the world. And when we think about what we know of how the pathogens or the virulence actions, as well as the spread of these diseases has changed over time. So what mechanisms specifically have changed, we can start to understand or target these different mechanisms for the outbreaks that are happening today, and be better prepared for them, as well as understanding if there's any sort of way to prevent them.
Yeah, you said that the syphilis and TB and other diseases have evolved with us. So what's that mean?
Yeah, so for tuberculosis in particular, we have tuberculosis being found in ancient Inca mummies from Peru, as well as ancient Egyptian mummies that date from about 400 BC and 3,000 years ago, roughly. So we also have a lot of cases of tuberculosis happening in that are associated with global trade, such as the Canadian fur trade. There's a great paper about that, and thinking about how wherever humans go, tuberculosis was always there. And we always have evidence for it. And when we base it on molecular dating, the molecular clock information also confirms that it's a very, very old disease, and that it's been slowly changing with and to different environments and populations.
You mentioned that not only have these pathogens evolved, but they've evolved with people. So, what does that mean? Have they become less virulent, more virulent? Are they infecting people in different ways that they didn't infect them before, or causing symptoms that are different? Like, what are some examples?
Yeah. Okay. Exactly. They've definitely changed over time. So when they enter new populations, those people may have different immune reactions to the specific strains of the diseases, or there could even be cultural practices in place that slow the disease, like social distancing. Or a good case for syphilis was that in Europe, because people were constantly wearing clothes and that it spread through flea-infected clothing, that it just jumped from clothing to clothing, and the fleas weren't bothered by it. So, but when it got into colder areas, it started needing the vector for syphilis needed different conditions to be able to basically jump from person to person. So it was slowed down by the temperature in colder areas, but it still was able to live in thatched roofs, on rats. It was fleas were able to live on like clothing that people had and were trading for, as well as like goods that people were, like spices or different foods that the rats and pests may eat. So the rat is really underrated in that it was a great vector. Like, it transported fleas and the disease all over the world, and we're very efficient at it.
Okay, interesting. I don't know. Have there been any big changes in these pathogens, or only small ones? And like, what are some of the time scales in which we've been able to observe changes in these pathogens? Like, do they tend to be quick or slow, and then quick all of a sudden, and then slow again? Like, what do you see?
Yeah, so it'll depend on the pathogen, honestly, and by chance a lot of the time. So when, for instance, it'll depend on what the pathogens are in contact with. Bacteria do this really handy thing where they do horizontal gene transfer, which is essentially they trade different parts of the genetic codes with each other that may or may not be beneficial to them. And by chance, a lot of the time, because they do it so often, they're able to gain new virulence factors, so new sequences of DNA that will make them more deadly, or affect the disease process potentially. And so when they're doing that with a lot, and they're in these perfect conditions, for instance, where there's high crowding for tuberculosis, there's going to be and it's humid and people are not being treated, it's very easy for tuberculosis or other bacteria to trade sort of genetic information with each other in these sort of unsanitary conditions. And so they can happen pretty quickly, but as far as like being instated, these virulence factors being seen worldwide, that can take a little bit of time. Yeah.
Have you been able to determine, what major factors cause an acceleration of the evolution of a certain pathogen?
Most of them are going to be in the transmission sort of routes and processes that happen. So if there's social conditions, for instance, where people are living in very crowded places, such as convents, tuberculosis can spread much more quickly and then become more virulent that way, just by the fact of getting in contact with different pathogens. But yeah.
For instance, like if you knew there was a major war, 800 years ago, does that tend to correlate with a sudden change in how a pathogen acts, or there's a sudden crowding or migration of a population from one place to another? Or, there was a mini ice age or an ice age 10,000 years ago, and certain pathogens appear to just disappear from the record, or that kind of stuff.
Yeah, and so, war is notorious for promoting all sorts of sicknesses and just pathogen transmission, because different people are coming into contact with foreign troops or foreign towns, etc. There's a lot of movement going on. So yeah, a lot of war and a lot of social things that can happen are very impactful for pathogens. And because a lot of pathogens like syphilis are reliant on human hosts, and they typically don't infect other organisms. There's plenty that do, however, but essentially with war, with many ice ages, and different events happening, they will facilitate different diseases. So you don't necessarily see different virulence factors coming up. You can, but there's generally what we see is going to be more and more cases occurring when there's favorable conditions for these pathogens.
Okay. Are there any big accelerants you've seen? Any trends or changes that cause a massive evolution or a super fast one of any given pathogen?
Well, the most recent, I would say the most spread of a disease I know of would be the most recent cholera outbreak in Haiti, where because we have global air travel, there was a health aid worker that went from Nepal to Haiti and transferred cholera overnight, essentially. But that also depended on the conditions that they were in. They didn't have separation of drinking and wastewater, which is what cholera really needs, is that fecal oral route to be accessible. And it was devastating. There's going to be the flu as well, that it changes every year. So that's got a really fast mutation rate, as well as it gets new virulent factors every year. And that's partially because of the number of strains that are out there. And we try to predict the most impactful strains every year. And that's why the vaccines for the flu are tailored to specific strains of the disease and try to prevent it. Yeah.
Huh. What are some of the big questions that you hope to learn with your research?
So when I'm really interested in understanding past human experiences with the disease and how that relates to the social conditions. So health is not universally felt across a population because there's minority groups, there's different living conditions depending on who you are. And most famously with the Mask of the Red Death and the plague, people were touched by the plague at different rates based on the socioeconomic status. And that doesn't necessarily mean that the disease progress within an individual changed, but those experiences, being able to have access to health and being able to maintain it, is privileged and also bioarchaeologically available to recover and reveal in the past. So I think it says a lot about personal experiences when you can assess someone's bones and molecularly assess their molecules to understand potentially what they died from or what contributed to their experiences. And what I'm really interested in is reconstructing like end-of-life circumstances from human remains in the past.
Oh. Have you yet been able to do that? Have you been able to get, I mean, I don't know, this stuff sounds kind of morbid and grisly to me, but I guess you have a fascination with it. So, I don't know, what interesting things have you discovered about people's, untimely or timely ends?
Well, I think looking at human remains, it's sometimes it's very obvious what someone died from, such as like blunt force trauma or like a bullet wound or something like that. But other times, like with diseases, it's less apparent or more nuanced. So I primarily work with human dental calculus molecularly, and that's like the tartar that your dentist scrapes off your teeth when you go in for a cleaning. That actually calcifies over time and becomes like a solid matrix, crystalline matrix that traps a whole lot of information about oral diseases and different conditions in the oral cavity. So we're not just limited by looking at cavities anymore. We can sort of reconstruct these oral microbiomes and different conditions of potential introduction of pathogens because the mouth is the largest orifice and most regularly used. There's a lot of potential for respiratory, gastrointestinal pathogens to be incorporated into dental calculus in the past.
Okay, interesting. So if you have someone's teeth that passed away, what are some new things that have been discovered? Any famous teeth that you've looked at and found interesting things about?
I don't think I've looked at anyone famous per se, but I do know that recent studies have found that I believe they were nuns. Don't quote me on that. These women, medieval women, have pigments in their teeth and in their calculus because they were painting illuminated books. And that is a very particular way to paint when you're constantly lip pointing your brushes and ingesting the paint and potentially toxic chemicals associated with them, which I mean, is interesting because nobody else in that society was doing, or very few people, I should say, were doing that sort of work. So it tells something about those individuals. And even further in the past, there was a recent study that basically found like chewing gum from the past and was able to reconstruct a lot of the pathogens and the seasonality that people were experiencing. And it's gross, but it's fascinating.
Oh, from chewing gum? What do you mean? What did they experience?
Well, so like seasonality, like they were able to find pollen, for instance, in this chewing cud that this person had. And so like in association with seasonality that they found, they also found like some immune factors being expressed and just sort of very personal information, I would say.
Huh. Interesting. Are there any particular bones that are better preserved than others? Maybe that, I don't know, I guess a funny analogy, bones being picked clean, but are there any bones that, maybe haven't been considered enough that could contain, a lot more juicy information than other bones?
Yeah. So human remains, like when it'll depend on whether or not you're a child or an adult. Child remains are very, very delicate. But then it also will depend on, for instance, whether or not you're buried or just left out in the sun, etc. That will also affect the information that you get from the remains, molecular or morphological information. But overall, it will depend on what your question is. For instance, a lot of population geneticists for human populations in the past will use the petrous bone, which is a part of the inner ear of your skull. And they use that to get human DNA from it to do population genetics. But you can also use the same element to do dietary reconstruction using isotopes. So it's also people doing pathogen research have regularly used the rib lesions, for instance, from tuberculosis individuals to recover Mycobacterium tuberculosis. And so, but you wouldn't necessarily use the same elements for different questions. Overall, people tend to use the petrous bone, really dense bone, so cortical bone, as well as human teeth, and sometimes just general lesions from the skeleton that are observable.
Oh, okay. Has anyone done a study on, I don't know, every possible bone that's been found to see, like, if certain bones will have, like you said, this particular one has some info, the teeth have another. I was thinking, what if a baby died? Do they tend to have less environmental information, but more information in their bones from their parents, from their mother, and just things like that? I just wonder what.
Yeah, and that's part of the exciting yet frustrating part of using human remains and skeletal elements is that it's sort of like we think of the preservation first when we are trying to decide what to sample, as well as the question that we're asking. But over time, throughout history, people have tried to identify the most ideal bone for X sort of analysis or that sort of thing. But people are really finding that it's going to be based on the question. So are you doing population genetics? Is there a petrous bone? Is there, was it preserved with shellac or anything else? Is there sand or other environmental contamination in there? Was it, particularly for museum collections, how heavily handled was the collection? And whether or not they were boiled or macerated in specific ways that may affect what we're finding in the molecules or the molecular data. But overall, it's a rather disappointing answer, but there's not one really good bone for any one research question.
Well, yeah, I'm not even assuming there's just one, but has there been a study, has there been enough study, you feel, of all the candidates to then line them up with all the things that might be learned from them? I don't even know what you'd call this, but again, has there been a big study of all the bones?
Yeah, people have tried to do a general overall DNA recoverability from different skeletal elements, which has directed several current studies even to really focus on the petrous bone rather than using the tibia, for instance, or even worrying about sampling the tarsal bones, for instance.
Okay. Is there a name for that? It's just, the study of bones, or is there something else, other name?
Well, so, the study of bones is osteology, and I think what you're asking about is the, optimization of the DNA recovery or molecular recovery from human remains, yeah? And that's generally a process that has been going on for quite some time since the advent of high-throughput molecular data technology.
Okay. So what do you see as the future of your work in the next few years? What do you think, where's there a breakthrough coming, or a new area or a new bone that, has a ton of study that needs to be done on it that's going to reveal a lot of cool stuff?
Yeah, so because I work, potentially I'm biased, but I do think that museum collections hold a lot of information that are just now being tapped in. And so there's a lot of information that is in there, including from non-human remains that could potentially inform how we think about and understand pathogens from avian sources or even from pigs, such as for influenza and other diseases, trying to get representative samples from different parts of the world and from different taxa. But I personally really, really like teeth. I think there's going to be a lot of work being done on human teeth as well as non-human teeth that really tell us about the organism as well as the lifeways and experiences of them, to try and understand what happened in the past.
Well, I guess the average age that people lived to wasn't very long, supposedly, 100 plus years ago, or certainly 1,000 years ago.
Mm-hmm.
So, I mean, maybe studying wisdom teeth, if you found the old skeleton, a couple thousand years old that had wisdom teeth, those teeth probably wouldn't have been exposed to the outer environment, to the food, et cetera, the person ate very long. They might have just gotten them before they died,
Potentially, yeah. And there's plenty of wisdom teeth
ones too,
Exactly. Yes. There's plenty of teeth, wisdom teeth, in the that are still in the crypts, for instance. And but then the issue becomes: do you destroy the jaw to get to them? Do you hope that they are developed enough to have enough dental pulp to sample, rather than just the enamel? And so there's just like those small details to consider, but definitely. Like looking at teeth as a way to time different development is already been done, but also doing that molecularly will be incredibly interesting.
Again, an erupted versus an unerupted tooth, they'd be exposed to very different environments. So, I wonder what you can learn from that. No. So there's a lot to see. Yeah.
Very cool. The uninterrupted teeth part is very cool because they if you've seen a child's skull with all the teeth forming inside cutaway jaws and skulls, it's almost like they're alien. So it's incredible. Like there's a full set of deciduous teeth, particularly when you're around 7 or 8, going on 10, and they'll have a full set like in their heads.
Wow. Well, very cool. Well, Rita, this is interesting stuff you're working on. I know obviously it fascinates you more, and some people may go, ugh, but it's cool that you're into this stuff. I mean, we need people interested in everything. So what's the best way for people to find out more about your work? And, have you set up your own exhibit, your own curated exhibit of things that you found over the years?
So probably the best way to find me at the moment is going to the Laboratories of Molecular Anthropology and Microbiome Research website through the University of Oklahoma. I should still be listed there. And I have worked with Dr. Courtney Hoffman. I also have worked pretty exclusively with the Smithsonian, the Natural History Museum. So I am available through them as well. But I don't have a website yet, which I need to get on. But
okay.
Yeah.
Very cool. Well, thanks for coming on the podcast. I appreciate it.
Thank you. It's been very exciting. Thank you.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.